which is Adam King from Waters. Please, Adam.
Hi there, hi there. Let me just share. Can everyone see that presentation?
We do.
Is it in presentations mode or is it in the right mode?
It's in presentation mode, but we can see the next slide, so you have to change screens.
There we go. Is that better?
Okay. Now is
okay.
Thank you.
Good afternoon, everyone. Good afternoon. My name's Adam King, and I am a senior scientist for Waters, based in just outside Manchester, and I work within the scientific operations department, focusing on metabolomics. So today I'm going to focus on eye mobility and how using eye mobility can help unravel some of the complexities that we all encounter within our metabolomic studies, and how that can help fully understand some of the molecules that we're looking at. So there's a series of different obstacles that we all encounter in our metabolomics experiments. These can be down to the fact that we're trying to analyze many analytes packed into a very small analytical space. Due to this, the many different analytes that we're trying to analyze that have various different responses, this can impact, have a broad dynamic range, which then obviously can be limited by the instrumentation's performance. Additionally, there's many isomeric forms of the compounds we're looking at, and obviously these possess some of the same masses, and as well as sometimes the same retention time information. So really understanding the impact of the isomers within our samples can be a challenge as well. And due to the many different analytes and different responses that they exhibit, and the impact on the instrumentation from chromatography to the detector, this can lead to poor reproducibility. And so these are all factors that could be contributing to issues that during our experiments. And finally, actually identifying the metabolites of interest and how you can actually improve confidence in what you're detecting. So let's look at a couple of examples here. Here we have couple of chromatograms from some extracted wood samples, specifically from xylem and phloem, and you can see from these chromatograms how complex and how many analytes that you are detecting within each of these runs. From just looking at the difference between these two different tissues, you can see that you can tell the difference quite easily, and building that into statistics, just looking at the features themselves, we can happily differentiate between these different tissues. But when it comes to actually looking at why these tissues are different and what molecules specifically, and identifying them, how they are driving that statistical difference, that is where the issues can come up. So if we zoom in on one of these chromatograms in the top left corner, this is one of the phloem extraction, and just look at a single peak within this chromatogram at 1.9 minutes. Below that, we have extracted the low collision energy spectra, so the precursor ions, from underneath that peak. And just taking the two largest ions within that peak, we can happily see these are two different molecules that are of interest, but they're both co-eluting at the same retention time. So if we wanted to ID these two compounds, looking at the high energy spectra after collision-induced association, because of them co-eluting, we end up having a more complex spectra, which basically just adds on like extra obstacles with trying to improve that confidence of IDing from theoretical fragmentation patterns. So how can we sort of look at addressing this complexity and deconvolving the information we're seeing? So there's a few ways that we can do this. The main way is trying to increase the peak capacity of our analytical system. So how can we go and increase the amount of peaks that we can detect and confidence in detecting those peaks. So we can go from the first step, which is the LC, and the first step of the separation. So we can move things from HPLC to UPLC, which will increase our peak capacity. We can look at doing multi-dimensional separations, looking at combining complementary chromatographic separations like helical and reverse phase to expand on the amount of analytes that we can see. And then we can also build in MS, the analyzer part, the MS dimension, and look at improving mass resolution, and also building in ion mobility as a technique to try and help with this. So combining all these things, your retention and UPLC, ion mobility, and improved resolution, you would end up building on this peak capacity that you can detect within your samples, and then improving the confidence of what you can actually detect. So, as I said, I'll be focusing more on the ion mobility portion of this system. So what really is ion mobility? Ion mobility provides separation of all the molecules based on their conformation, their size and their shape. So this in itself is an orthogonal separation to your chromatographic separation, the mass separation, and it allows you the ability to see more. It also has the ability to resolve some isomers depending on their size and their structure, and therefore, because you are separating by an additional dimension, you can then obtain cleaner spectra to help with identification. Alongside that, each molecule can have a collisional cross-section calculated, and this is independent of matrix, independent of the system, and it's specific to the compound. So this is involved with calibrating the ion mobility cell on the instrument, and it can improve confidence. We can build libraries and search libraries of CCS values to improve that confidence in identifying that compound. So our typical ion mobility separation on our Synapt XS instrument involves this T-wave system here. So starting from the left, you can see your chromatographic peaks come in, and the red and the blue dot are two co-eluting ions that you can't see from the chromatography. These then enter the T-wave cell, and through the ion mobility cell in the middle here, they get separated out. These then can get passed through from a drift time separation into the mass analyzer to have to be analyzed. So combining this with collision-induced association, you can then switch to another function that these two co-eluting species are separated in the ion mobility cell, fragmented, and then these fragments with their precursors in the previous function are time aligned, so then you've got that additional separation and the clearer spectra with the fragments now associated with a time separated precursor. So with all this together, with the mass accuracy of the instrumentation, retention time, these calculated CCS values, and the fragmentation, it can dramatically improve the confidence in the IDs that you are generating. And putting this data into some of our software Progenesis, it can take into account the CCS values and the separation, and can, as you can see here, if we move back to the wood samples, that taking those two peaks, that one peak which had those two different ions underneath there, the software's managed to reduce the amount of fragment ions that were complicating the high energy spectra, allowing us to have better confidence in the IDs that we have here. The first one of the peaks for corresponding to 309 has been found to be linked to salicin, and you can see that the red peaks are matched fragments based on their theoretical mass. Below we have an indole acrylic acid, which has got a cleaner spectra and a lot of matched theoretical fragments there. So we can see how mobility has helped along that additional dimension with cleaning up the spectra. So taking another example, we have some analysis of certain wines that was performed by an ex-colleague of mine. At the bottom left, we have a peak at 1.75 minutes that again has some co-eluting features. And if you can see the arrows pointing to above to different spectras, they look very similar. And obviously quite difficult to differentiate which ions correspond to which precursor ions. But moving across to the top right of the screen, we have a mobilogram showing these two precursor ions being separated by drift time and possessing different CCS values. So you have one at 227.18 and one at 255.93. So from this and looking at their drift time separation, we can produce much cleaner spectra, which has allowed these two different molecules to be identified as cysteine A, which is produced during the fermentation process in wines, and a flavonoid. So that's already shown about how improving that confidence that you can have from ion mobility just by cleaning up that spectra and allowing you to more accurately perform your assignment of analytes. But how do we deal with isomers? Some of these smaller isomers, they can't be separated by retention, and they struggle to be at similar masses, so you can't separate them by mass. So what do we do with them? So we know that a lot of structure-related isomers can have different biological processes and be affected by the biological system in different ways. So it's important to be able to differentiate between them. So even with ion mobility, a lot of the conventional mobility cells don't provide the adequate resolution to separate them. On the right, we've got a couple of examples of molecules and isomers that we want to look about separating. So at the top, we've got a couple of bile acids, and below we have a couple of methyl histidines. So how can we go about separating them? So one of our newer instruments, mobility-based instruments, is the cyclic IMaSS, the Select Series cyclic IMaSS instrument. This instrument has changed. If I can get the pointer, can I get the pointer? Right, yes. So this instrument has altered the linear ion mobility cell that we previously saw in the other slide and replaced it with this cyclic mobility cell. So pretty much the ability for this is to inject ions into this cyclic device and send them round as many times as we like. And this in itself allows for improved resolution and separation based on the molecules traveling through the mobility cell. It allows us to optimize this even further by separating and then fragmenting. We can move things in and out, back and forth in this cyclic device to cut out resolved peaks and then re-inject them into the cyclic device to also improve additional separation. So realistically, what does that mean? So here's a graph just showing a couple of peptides that we couldn't resolve based on the original linear ion mobility cell. And you can see over time, with performing multiple passes around that cyclic device, that we've managed to achieve increased resolution on the instrument to actually fully resolve these two peptides. And on our previous generation of instruments, we weren't achieving anywhere near the level of resolution required to separate out these two peptides. So peptides obviously are larger, a lot easier to separate. So what this is really translate to those compounds that I was showing at the beginning. So here we can see at the top the two bile acids, the mobilogram showing how we've managed to almost get baseline resolution on these compounds after 20 passes around the cyclic mobility cell. So this separation was achieved in 200 milliseconds, and achieved a resolution of 290. So we can now confidently differentiate between these two bile acids, and if they have different biological effects, we can now identify that. And below, in this other mobilogram, we can see the methyl histidines have had baseline resolution after 5 passes, which was achieved in under 50 milliseconds, and achieved an IMaSS resolution of 145. So this has shown how we can now differentiate between these different isomers quite easily. And just to confirm, once these have gone through and had their multiple passes from the cycler device, we can perform collision-induced association on them, and we can produce different spectra for these bile acids to help with IDing processes. And the same with methyl histidines. These were confirmed with standards to make sure that we could actually tell what arrival time each of these methyl histidines were experiencing. So, in summary, iMobility can increase this analytical peak capacity, which can improve your confidence in what you're seeing and make sure that there's nothing hidden within your sample. It can provide CCS values to help with identifying specific compounds, and that can be related to online libraries, experimentally, as well as theoretically in silico CCS values calculated. It can separate co-eluting ions, especially if we're wanting to try and work out which one is actually an important isomer. And using the cyclic ion mobility instrument, we can confidently resolve structural isomers that we weren't able to before. So just some acknowledgements of people that we worked with here, and thank you very much for listening. And if there's any questions, I'll happily answer them.
Okay, thank you very much, Adam, for your presentation.